Now that we've seen a part of the course that's called the chemistry of life and also the biological molecules, where we explored the structures of sugars, lipids, proteins, and finally nucleic acids, I think it's time to put all of this together and start to explore something called the tour of the cell. So basically in this chapter we will look at different kinds of cells, but most of all, especially at the beginning, we will define what a cell is and how the cell is the fundamental unit of life. It all starts with a cell. So some cells, some organisms, I should say, are either single-celled or they can be multi-celled or multicellular. So as you can tell here, some cells might work by themselves. They might live by themselves and live a life that does not involve the formation of a major structure with other cells, or they can assemble with one another and form a more substantial structure where the cells now depend on one another to survive. So in the case of plants, many animals, and most fungi, cells will form subunits. They will organize themselves and then start interacting with one another, like this here. We'll have some interactions between all of those, and they will form a bigger subunit, much bigger than a single cell. So a single cell would be something like this, and we would have many, maybe single cells in a certain medium, but those single cells might not really interact with one another. And by interaction, I don't mean that they don't quote unquote talk to each other. They do communicate, but their survival is not dependent on their interaction with one another. So as you can see here, I'm showing many cells that are connected with one another, and they form this bigger substructure, and this is seen in multicellular organisms. So when you think of cell theory says a few things, and it says that, well, all living things are composed of cells. So like I said earlier, basically the cells are the basic, most simple living structure of any living thing, whether it's multicellular or only single cell, monocellular. And the other thing is that all cells come from another cell. So this is a kind of like chicken and egg type of thing where we don't know who started the first thing, right? Is it the chicken that had the egg and then gave a chicken and then they gave an egg? Well, who started? Is it the chicken or the egg, right? Well, we know that all cells come from another cell. And when we explore cell reproduction, we will talk about that. Our goal at this point is not so much to look at cell reproduction, but basically I want to give you an understanding of what cells are. And I could not explain this before I show you a scale to demonstrate how big or small cells are. So I just want to attract your attention here on that scale right here, and then we have units of measurements, right? So it's a distance, for example, right? So we are very familiar with millimeters, we are familiar with meters. Well, when you subdivide those millimeters into thousands of millimeters, we have the micron. Okay, so a micron is 1,000 times smaller than a millimeter. And if we subdivide this micron into even smaller subunits, then we end up with a nanometer. and the nanometer is 1,000 times as small as the micron, and nanometer is 1,000,000 times as small as the millimeter, and it is 1,000,000,000 times as small as the meter, right? So a nanometer is very, very, very small. Now, if we wanted to see those structures, well, there's no way with the naked eye, there's no way we could see any atoms. We could not see any proteins, we could not see any lipids, and certainly we could not see any bacteria. In order to see those small structures here on that side here, you need at least an electron microscope. Now, when you want to start to visualize structures, cellular structures, including cells themselves, and what we have here, we have an animal cell, and we also have a plant cell here. Well, what you need in order to visualize those cells at the individual level, you need at least a light microscope. And in this class, we will absolutely work and learn how to use light microscopes. It's quite interesting, and it's quite revealing when you can see the cells. Now, as we move further and further, we can tell that some structures we can see with the naked eye. So if I put in front of you a human egg, you could, in theory, see it. It's very small, but still it is possible to see this with the naked eye. A frog egg is much bigger, and of course we know the chicken and ostrich eggs, right? So what I want to give you here is just an understanding that we will be working and understanding and studying cells, and you will not necessarily see those cells all the time. You'll be able to measure what they do, but you won't always be able to see them. So now when we explore the world of cells, we know that there are two major categories of cells. The first one is something that we call prokaryotic cells, and then the second category is what we call eukaryotic cells, right? Prokaryotic cells versus eukaryotic cells. So when we're thinking about bacteria, archaea, we know that those are prokaryotic cells. So everything that has to do with bacteria is a prokaryote. When we look at plants, when we look at protists, very small aquatic animals, and when we look, of course, at bigger, higher animals, we know that those the cells of protists, plants, fungi, and animals are eukaryotic cells. Now, what I want to show here is that basically when you look at a eukaryote versus a prokaryote, there are very important features that are there. We will explore many of those features, but there are features that are not there. And one very important example is probably the most widely known difference between eukaryotes and prokaryotes is the presence of a membrane enclosed nucleus. So the nucleus is what we find inside the cell. So if I have a cell that's about like this, what we have inside, if we look at it through the microscope, we will see a structure like this. And what we have in the nucleus is DNA, right? So we would have the strands of DNA inside of that nucleus here. Well, this is found only in eukaryotic cells. Prokaryotic cells do not have a membrane enclosed nucleus. So basically their DNA that we can see here is structured in such a way that doesn't need to be contained inside a membrane. That is the main difference. There are other differences, but that is the main difference between prokaryotes and eukaryotes. So when we think of the two major categories of cells, there are some differences, and I showed one of the difference. Basically, the nuclear membrane is a very important difference between the two of them. But there are things that are common, and what's common is that each cell, whether they're prokaryotic or eukaryotic, are bound by a thin plasma membrane. And we will see more in detail what a plasma membrane is. But both types of cells have a plasma membrane. So the plasma membrane would be something like this. We would have if we looked at it under the microscope, we would see the cells as single units, and those units are contained by a plasma membrane. So basically what we would see here would be the plasma membrane right there. Inside the cell, so what the plasma membrane, it creates an enclosure for what's on the inside. And what is on the inside is known to be the cytosol. That's what we have here. So this is where we have all the smaller particles inside the cell. This is where it's like. And it's mostly made of water. It's a little bit thicker than water, like we can even say like kind of like jelly-like fluid. I'm not sure if this is a very, very accurate description of it, but what I wanted to show here is that it's not really water because there's many components in it, and those components are suspended, and of course this will affect the viscosity of the contents. Now, another very important similarity of the two types of cells, something that they both have, is that they both have DNA, right? We've seen what DNA is. We've seen that it's made of nucleotides that are interacting with one another via hydrogen bonds and all of that. Well, when this DNA contains many nucleotides connected to one another, and then it creates this long strand. Those strands are now known to be chromosomes, and those chromosomes are found inside the cell. Now, both prokaryotic cells and eukaryotic cells have chromosomes, but just remember that eukaryotic cells also have a nuclear membrane around those chromosomes to separate these chromosomes from whatever is found inside the cytosol. Now, what I want you to do is to pause this right now and then just go back a few seconds and replay this. This is important because I link chromosomes, cytosol, the nuclear membrane, and all of that, so I think that's important to do. Now, another kind of structure that we will find suspended inside the cytosol is ribosomes. So ribosomes are tiny factories, tiny little machines that will convey and transform the information found in DNA. So we have DNA, and from DNA we can create proteins, as we've seen before, right? So those proteins are encoded by DNA, and ribosomes are the wonderful little machines that allow this part of this process to happen. So this picture here shows a typical eukaryotic cell, and I just want to make sure that we understand what I mean by typical eukaryotic cell. What it means here is that not all eukaryotic cells look the same. Some of them have different features than others. When I say typical, it's because we want to, I just want to make sure that I show you a good representation of what a eukaryotic cell would be. So as an example, when you see here the flagellum right here, the structure right here, well, only a few cells actually do have that. Sperm cells, for example, do have a flagellum to help them swim, but not all cells have that. So this is what I mean by that. In addition, when we look at different tissues, so for example, cells flowing in your blood right now. They're called lymphocytes, and those lymphocytes have a very typical shape. They're usually round with a big, huge nucleus. When you look at the cells lining your gut, those cells they're all organized. They're basically structured like a little fence connected to one another, smaller nucleus. They have a very specific border to them and all of that. So that's why we say typical here. Now, remember that the difference between eukaryotic cells and prokaryotic cells is the fact that eukaryotic cells do have a nucleus, and this nucleus will protect the DNA inside of that cell, right? So it will make sure that the DNA is kept in only one portion of that cell. Now, the other thing about eukaryotic cell is that you will find when we look at it, and we will explore many of those little smaller structures here, is we'll see that inside that whole cell in the cytosol, we will be able to identify many different organelles. Organelles stand for like smaller organs, if we can say it like that. And some of those organelles accomplish very, very specific tasks within a cell. Well, prokaryotic cells don't really have organelles inside of them, and one of the reasons for that is that there is not a lot of compartments found inside a bacterium, for example, which is a prokaryote. The eukaryotic cells will create a lot of compartments within the cell. So this is why we have all of those smaller organelles right here. So you see peroxisome, we have lysosome, we have mitochondrion, we have Golgi vesicles right here, we have endoplasmic reticulum, some of it is rough, some of it is smooth, and all of that, right? So the goal of this present slide is just to demonstrate the complexity that we have when we look at eukaryotic cells. So when we look at eukaryotic cells, we also want to make sure that we make a difference between animal cells and plant cells. We want to make sure that we can see the difference between the two of them. Now, the similarities are multiple. There's some of the plant cells really do have mitochondrion, peroxisome, Golgi vesicles, as we've seen, rough endoplasmic reticulum, there's smooth endoplasmic reticulum, and all of that. So, and please don't forget that if you're struggling with this right now and thinking, oh my God, I don't know what a rough endoplasmic reticulum is and Golgi vesicle and all that, don't worry about it. What I want to do right now is just to demonstrate the differences and similarities, right? But one thing that is found in eukaryotic cells, but in plant eukaryotic cells, we have those very important parts here. And this one, the chloroplast that we find here, this little green particle, an organelle right there, is responsible for the process of photosynthesis. The reason why plants can incorporate carbon dioxide and transform that into a plant, a living thing, and leaves and wood and all of that is done through photosynthesis. And photosynthesis is possible because of the chloroplasts, right? What we have as well is vacuoles. We'll see, and we will have a lab where we will be able to observe some of those vacuoles inside of the cell. So don't forget, here it's not so much about the details and understanding everything what the organelles do. We have a special section for that. What I wanted to show here is just to give you an overview of the eukaryotic cells. One thing that I want to describe now is a feature that is common to all types of cells, whether they're prokaryotic cells, eukaryotic cells, or even in eukaryotic cells, even though they might be plant or animal cells. All of these cells have this very important common feature, and this is known as the cell membrane. So the cell membrane is something that's found around the cell, right? So when we look at a cell like this, and like I explained before, whether it's eukaryotic or prokaryotic, it doesn't matter. But the cell membrane here, what keeps the fluid, which is mostly water, keeps it inside this barrier here. It forms a barrier, and the water that's found inside the cell, also known as cytosol, is different from the water that's outside the cell. So what we have here, we have inside the cell, and all around here is outside the cell, right? So the water, or mostly what is dissolved in that water outside of the cell, is different than what we find inside the cell. And this is very, very important for cells. We need often to keep whatever is inside the cell different from what is found outside of the cell right here, right? So what is out doesn't need to come in, and what is in doesn't necessarily need to go out either. So we need to keep a difference between what's found inside the cell and what's found outside of that cell. So now let's say that I look at the cell membrane. So what I have here, I'm drawing a cell membrane. And let's say that I take this little piece of the cell membrane here, and I expand it, right? So I look at it closer. So what I would see right here in this part right here is I would see that there are a number of lipids that compose this membrane. So the lipids, as if you remember well, the lipids have those tails like this, right? There's like those zigzagging tails like that, and some of those tails are saturated as we see here, but some of those tails might be unsaturated, and because there's a double bond here, we have this kink that happened here, right? So this part here is the lipid part of that cell membrane here. Now, when we look at the kind of lipids we find in the cell membrane, we find this very, very important and different type of lipid, and those are called phospholipids. And the reason why they're called phospholipids is because they do have a structure like a lipid like this, so this zigzag part, but also they also have something right here, and I'm not going to draw the exact molecule here, but what we have here is something that we call a polar head. Polar, P-O-L-A-R, head. And that polar head is polar because it is charged, and usually it is charged negatively, because if we're talking about a phospholipid, well, you can imagine that what we have in this head are PO4 minus, or phosphates. What happens is this: when we have a phospholipid, we usually have a phosphate as the head, or included in the head of that lipid, but also we have those chains right here, chains right here. So you see there's one chain here, there's another one, and both of those chains are attached to the polar head, which confers this very interesting property. Because this part here is polar, it does interact with water very well. Water will come close to this and interact and form hydrogen bonds with this here. But this part here, that is the lipid part, we've learned that lipids are hydrophobic. So water, when it comes to interacting with this part of the molecule here, of course, it will not interact. So you can imagine that water will have to kind of like interact with part of the molecule, but not with the other molecule. And there's a name for a substance like this. This substance is called amphiphile, meaning that it can interact with water, but there's also a portion of it that's hydrophobic that prevents this interaction with water. Now, because of the amphiphilic properties of phospholipids, and and because we need to have this membrane needs to interact with water on one side and then with water on the other side, right? We have water inside the cell, the cytosol, and we have water on the outside. Well, those lipids, those phospholipids, will need to interact with one another in order to allow this interaction with water. So here's the way it's done, right? So what we have here is we will have our phospholipids. So I'm just drawing the heads of the phospholipids here. I'm just drawing a few of them. You can imagine, of course, there's billions of them. And this is the lipid part of each of those phospholipids. Here we have all of this, and now for sake of simplicity I'm just going to draw straight lines here. We understand that these are, if I draw a straight line, that's not really quite the representation, it's not the reality, but you know, I don't want to spend half an hour just drawing those structures here. So on one side of the membrane we will have those phospholipids right here. So interaction with water is possible right here at the surface of the phospholipids, but on the inside it's impossible. Now this here would be the water that's outside, right? So what I want here is to show that this is the same water we have here. Now when we want to create a membrane, and we also need to interact with the water that's in the inside, and the properties of these phospholipids will allow for the structure to be made where we have phospholipids like this facing each other through the lipid portion of it, or the hydrophobic portion of it, and then water on the inside of the cell. So we are now inside the cell. So this is the water that's right here. This water will be able to interact with the heads here, but this leaves us with an interesting part here of the membrane, and this part here is highly hydrophobic. So because of the structure of this membrane, and that membrane here is now known as a phospholipid bilayer. Phospholipid bilayer. Why bilayer? Because of two. There's two layers. The layer number 1, which faces out, and the layer number 2, which faces in. And of course, there's water on the outside where we have ions that can be dissolved. So I drew potassium, sodium, chloride, and all that. There's of course other ions that can be dissolved, and then we also have water on the inside where other ions can be dissolved, and like I drew sodium here. Now let's say that doesn't exist. What I'm going to tell you doesn't exist, but just let's pretend, right? Let's pretend that inside the cell here there's no chloride and there's no potassium, but there is potassium and chloride found on the outside, as we've seen here. Do you think these ions will be able to to go through the membrane and from the outside in, right? Do you think that's going to happen? Well, pause this video, think about it for 2 seconds. So now that you thought about it, you probably came up with the answer, well, no, because although it might interact with the head here because this is hydrophilic, there is this region here all inside of that membrane, the inside here, that is totally hydrophobic, and all water will be pushed out. There's no water that can interact with this part here, and of course if there's no water, there's no way we can dissolve ions in it. So ions cannot, by themselves, they cannot cross this membrane here. So whatever ion is on the outside will remain outside unless there's a special door for it. There's a special passage that allows those ions. So now enough with my drawings. I'm going to show you something more useful, I suppose. So here's what we have here. We have the cell, and this is your eukaryotic cell. We know that because there is a nucleus, right? And what we have here is a cytoplasm on the inside, and also known as cytosol. And the cytosol we know contains water, and of course we have the extracellular fluid, which means is another word to say, well, this is the water and everything that's dissolved in it, water and everything, all the ions and proteins and everything, that is found on the outside of the cell. Now, if you look at it, we have this nice little rectangle here, and then we blow up this rectangle, and what we see here is the cell membrane of that, right? So what we see here, if we could explore this, so we'll take that section here in green, and then we will blow it up again. And so basically what we're doing is just amplifying the image here. And we see that in red, right here, we have the polar heads of the lipids, right? So we see the polar heads right here. And on the inside here in yellow, we see that we have the lipid portions of those phospholipids, right? So basically, when we look at the phospholipid here, we amplify this, we see that there's a hydrophilic head right here that contains a phosphate. The phosphate is right here. We can see it very well. That's hydrophilic, which means that it can interact with water. And then there's also a hydrophobic tail right here. Actually, there's two of them. There's one here, and of course there's another one here. And I attract your attention. I just want to make sure that you understand what that is here. You see there's that kink right here. Well, that's because there's an unsaturated fat right here, which allows for the creation of a kink like this, basically because there's a double bond between two carbons, right? So when we look at the structure of those phospholipids, we see that the polar head is right here, and the way those phospholipids will organize themselves in a phospholipid bilayer is that everything that needs to interact with water will be opposed to one another. So the polar heads will be on the outside here, and the polar head will be on the inside here. Remember that we have water on both sides, and then whatever is found in the middle part here will contain absolutely no water. The other thing that's very important, and I did not mention it so far, but I really need to mention this, is this. What we have inside a cell membrane, the cell membrane is way more complex than just a phospholipid bilayer. When you look at the cell membrane, there's also all sorts of proteins and sugars that are found inside that membrane. Remember when I told you that there was, like, say, a potassium ion that could not by itself make its way on the inside? And I said, well, it won't by itself, but if there is a door for it, maybe it will. Well, you see here, sometimes cells in their membranes, they do have a channel, and that channel can be specific for different types of molecules. So say that we have a small ion of potassium right here. Maybe that small potassium ion will be able to make its way through this channel and then travel from the outside to the inside of that cell right here. So remember this. The cell membrane is way more complex than just the phospholipids. Yes, there are an amazing number of phospholipids in the bilayer, but there's also a lot of other molecules are embedded in this membrane, including proteins. Some of them can form channels. Some of them are just embedded through and through that membrane here, and we can see this very well here. And sometimes some of those proteins will adopt a very specific structure in order to be able to go through that membrane. So what we see here, if we look right here, we see that there is, I just don't have the right color, so let me just circle this again. What we see here, you see you have this very interesting structure here. It's called a alpha helix protein. Wow, remember that, right? Remember that secondary structures are very important. Sometimes proteins will adopt some of those secondary structure, and a alpha helix is something that looks like this, right? We have the strand of protein of amino acids, the primary sequence that can turn on itself and form this structure, and will be stabilized by forming those very interesting and important hydrogen bonds between them. So this whole structure here, which is the alpha helix, will be stabilized like that. Now, I'm digressing right now. I'm going way out. I shouldn't be there. What I want to show and what I want to say is just that the cell membrane is made of lipids, but also all sorts of other things, including carbohydrates, including proteins, including glycolipids, and of course including phospholipids.